Hello, everyone. Welcome, and thank you for joining the Interventional Psychiatry Consortium. My name is Yaakov Green. I'm a psychiatry resident at Columbia and one of the organizers of the consortium, alongside Ailani, Jeff Zabinski, and Jarrod, Ehrie, as well as our advisory board. For those of you joining for the first time, the consortium is an educational network bringing together trainees and other advanced learners together for a shared educational experience in the rapidly growing field of interventional psychiatry and neuromodulation. We now have 372 members from 63 institutions from around the world. Last time I checked, 11 countries. Thank you for being part of this. And this reflects the need for education in this space. Each year, we will host a series of expert-led talks that highlight the latest advances while staying grounded in clinical practice. And we'll soon add other formats and potentially community building events too. As for the format today, we'll soon hear from our speaker. Then we'll have a Q&A, during which time you can use the chat to ask a question or raise your hand to be unmuted. The sessions are recorded and will eventually be made available to you all. Today is the second talk in a phenomenal lineup of speakers covering a variety of topics. If you haven't already, take the chance to sign up using the link that was sent to your emails. Now, I am thrilled to introduce our speaker. We are so incredibly lucky to have Sarah Hollingsworth Lisanby with us today. She is truly one of the pioneers of this field. Dr. Lisanby is currently the founding dean and foundation professor of the Arizona State University School of Medicine and Advanced Medical Engineering, and professor emeritus at the Duke University Department of Psychiatry and Behavioral Sciences. Prior to her current role at ASU, Dr. Lisanby served as the founding director of the National Institute of Mental Health Division of Translational Research. She played a leading role in the NIH Brain Initiative, having co-led the team focused on large-scale neural recording and modulation devices, and co-led the Brain Behavior Quantification and Synchronization Program. Formerly chair of the Duke Department of Psychiatry and Behavioral Science, and J.B. Gibbons Endowed Professor with tenure, she founded and directed both the Duke University and the Columbia University divisions of brain stimulation. She also founded and directed the NIMH Non-Invasive Neuromodulation Unit, an intramural research lab specializing in the development and first-in-human testing of novel brain stimulation devices. An accomplished clinical translational scientist, Dr. Lisanby has authored over 300 scientific publications. She is internationally known for her groundbreaking research supporting the FDA approval of novel devices for the treatment of depression, including transcranial magnetic stimulation, which we'll hear about in a future talk, and vagus nerve stimulation, which we'll also hear about in a future talk. She also pioneered magnetic seizure therapy, one of the topics of today's talk, which we will hear about shortly, as a rapid-acting antidepressant from the stages of device development, preclinical testing, first-in-human trials, and multi-center trials, leading up to the pivotal non-inferiority trial against gold standard treatment designed to support FDA approval. Dr. Lisanby serves on the FDA Neurological Devices Advisory Panel and has led key leadership positions in professional organizations, including president of the International Society for ECT and Neurostimulation, and chair of the American Psychiatric Association Task Force to revise the practice on electroconvulsive therapy. Dr. Lisanby serves on numerous national and international scientific advisory boards as well. She's a board-certified psychiatrist and Distinguished Life Fellow of the American Psychiatric Association. Now, without further ado, Dr. Lisanby.
Thank you so much for that kind introduction and for the opportunity to address this group about electroconvulsive therapy and magnetic seizure therapy, two of my favorite topics. Our learning objectives for our talk are to go over some basic introduction to these two interventions. I'll go over some fundamentals of dosing for seizure therapies. I'll introduce you to some conceptual models for how seizure therapies may exert their clinical effects, and we'll wrap up with some future directions that may help inform mechanisms of action and new approaches to optimizing dosing. So let's get started. ECT. It's the induction of a seizure under generalized anesthesia and muscle relaxant using an alternating electrical current applied through scalp electrodes. We typically give treatments 3 times per week for a series of weeks. It starts with a device, which is a pulse generator, which delivers bidirectional pulses, trains of pulses via scalp electrodes. These induce electrical fields in the brain that in turn induce generalized tonic-clonic seizures that then have lasting changes in brain function. ECT has an exceptionally broad therapeutic spectrum, meaning it works in a wide range of conditions, not just depression. It also, though, has a fairly narrow therapeutic index, which is the separation between the dose that's effective in treating depression, so you can see here percent remitters and responders to a course of ECT, and here is the impact on memory. So on the y-axis is autobiographical memory score from before to after ECT. What we want to do is find a dose that optimizes that separation between what's effective and what causes side effects. Now, importantly, these clinical effects are dose-related. So let's start by defining what do we mean by dose of ECT. So the key components of dose start with electrode placement. It's literally where you place the electrodes on the head. 3 common electrode placements in clinical use are bilateral temporal, right unilateral, and bifrontal. When you move where the electrodes are placed on the head, what you're doing is you're moving the distribution of the electric field in the brain, which we can simulate using electric field modeling shown here. Electrode placement has a powerful impact on clinical outcome. As you can see in one of our early studies, on the y-axis is amnesia score for 2 different types of memory, personal memories and impersonal memories. The blue bars show the amnesia score with bitemporal ECT, and the red bars show the amnesia score with unilateral ECT. So you can see there's a dramatic reduction in amnesia when you move from bitemporal to unilateral. There's also significant change in the distribution of the electric field between these 2 electrode placements. This clinical result highlights the importance of spatial targeting in terms of clinical outcomes. The second key element of dose is pulse width. The ECT pulses are square waves, and we can measure their width in milliseconds and their height, that's the current, in milliamps. The strength duration curve shown here depicts the relationship between the pulse width on the x-axis and the current amplitude on the y-axis. The line shows you the minimum current needed at each pulse width to induce an action potential in a mammalian neuron. Ultra-brief pulse width is defined as being 0.25 to 0.3 milliseconds, whereas brief pulse is longer, defined as 1 to 2 milliseconds. You can see that the ultra-brief pulse is closer to the knee in that strength duration curve, which is called the Cron axis. That means it's closer to the most efficient part of this strength duration curve. Pulse width is a powerful driver of clinical outcome. As you can see here in this study on the y-axis is amnesia score with standard pulse width in the dark red and blue, which corresponds to the 2 different electrode placements, and then the lighter pink and light blue is the ultra-brief versions of those. So you can see when you shorten the pulse width, you're significantly reducing the amnesia score. This tells us the importance of temporal tuning. So we've got 2 concepts, spatial targeting, temporal tuning. But why are briefer pulses safer cognitively? We know that these briefer pulses can induce seizures at lower total charge. Here you can see we've titrated the seizure threshold with an ultra-brief pulse and a standard pulse for the same electrode placement. Now, why might this be the case? There's some theories, we've posited some of these, that the standard pulse widths have a different, that there's a different time constant between soma in the dotted line and so the cell body, and the axons in the dark, in the solid line. So there's a different ratio with standard pulse width versus ultra-brief pulse width in terms of which part of the neuron you're actually stimulating based on their different time constants. A third element of defining dose is seizure threshold, and we titrate this. The conventional way of titrating seizure threshold is by giving trains of pulses with increasing duration and frequency until a seizure is induced, and then you set the clinical dose as an increment above that. However, spoiler alert, there's a better way to account for the anatomical variation and individual parameter contributions. We're going to come back to that at the end of the talk, but I'm showing you here what is done standardly in clinical practice. The reason why we think that there may be a better way to do this starts with our rethinking of how we define dose with ECT to begin with. I'm showing you here one of our papers which demonstrated that each electrical parameter, so the pulse width, the frequency, the train duration, the pulse wave, and all these train characteristics, have independent contributions in terms of the physiological impact. You can't just sum them all up and think that they're all created equally. This chart reflects what is the current break-even point between efficacy and side effects. It shows you that 6 times seizure threshold with the right unilateral electroplacement using an ultra-brief pulse width is able to match the efficacy of bilateral ECT, the green bars are efficacy, while having less amnesia, that's the red bar, than the bilateral. So it means sticking with the ultra-brief pulse width, going 6 times the seizure threshold with the unilateral. That's the current best way that we know of to balance efficacy and side effects with conventional ECT. So I'm here highlighting that here. We tested the efficacy of this ultra-brief pulse 6 times threshold right unilateral ECT in over 200 patients who were had depression, who were all elderly patients, and we found significant response remission rate. On the y-axis is the percent of patients achieving response after each individual ECT session on the x-axis. The orange bars show you the cumulative percentage of patients achieving response. Half of them responded in 2 weeks, 3/4 responded by 3 weeks. In this study, our remission rate was 83% among completers. Now, after responding to a course of ECT, it's very important to put in place a relapse prevention strategy. Otherwise, the depression can come back. You're looking at here a survival curve comparing 2 relapse prevention strategies in this prolonging remission depressed elders study. In the green line are those that were on medication alone, and the orange line are those that received a combination pharmacotherapy along with a symptom titrated longitudinal ECT, which means we continued the ECT on a continuation and maintenance schedule based on the reemergence of symptoms. So it was an individualized schedule. Now, I've talked about that therapeutic index of ECT, and all of the doses of ECT that are effective therapeutically also have some risk of side effects. So we'd like to re-engineer ECT to get rid of that side effect burden. We'd like to be able to achieve the unparalleled efficacy of ECT without the amnesia. That would truly be the holy grail. That's been the quest of my career and many others' careers. Our approach to accomplishing this goal is to ask, well, how does ECT work to begin with? A greater knowledge of mechanisms could inform how we would engineer ECT to retain its efficacy while paring away its side effects. So in the next part of the talk, we're going to unpack ECT and rebuild it such that it's better, faster, and safer. And now you know my taste in movies and TV. So let's unpack ECT. Back to the multi-step process, starting with the device, the train of pulses, the scalp electrodes, the E-field, and seizure. Let's zero in on the E-field and the seizure. We don't know whether it's the electric field or the seizure or both that drive the efficacy and side effects of ECT. So unpacking these mechanisms may be key to optimizing the efficacy, the safety of this highly effective intervention. But spoiler alert, this slide is incomplete. There's a novel mechanism that has been identified that needs study. We're going to come back to that novel mechanism at the end of the talk. But first, let me present a conceptual model for how ECT might work, considering the E-field and the seizure. So we've got the E-field, we have the seizure, then we have these 2 types of clinical outcomes, cognitive side effects and antidepressant efficacy. Well, one model says that the E-field drives cognitive side effects while the seizure drives efficacy. But you could posit the opposite, that the E-field drives efficacy while the seizure drives side effects. I call these models A and B. Or it could be the interaction, something about the interaction of continuing to stimulate while the brain is seizing that might result in these clinical outcomes. Now, to test these models, it would be helpful to have tools that would just apply E-field or just apply seizure so we could unpack their independent contributions. Unfortunately, we can't do that with conventional ECT. Here's why. So again, here's the E-field simulation of the 3 conventional ECT electrode placements. They all induce strong diffuse electric fields, and they all, while also inducing a seizure. So you're getting both at the same time. They're not uncoupled. That's part of why we developed magnetic seizure therapy. What magnetic seizure therapy does is it induces a generalized seizure with a minimum electric field induced in the brain using a specialized transcranial magnetic stimulator. So it's a weak E field, but inducing a seizure. Now, on the right-hand side, we're looking at non-seizure inducing interventions that deliver E fields. TEST stands for transcranial electrical stimulation therapy. It applies diffuse electric fields that are modeled after bifrontal ECT, but we stop short of inducing a seizure. This allows us to ask, what is the E field doing on its own without seizure? And then the lower right-hand part of the slide is, of course, transcranial magnetic stimulation, which induces weak electric fields without inducing a seizure. And we already know that that's antidepressant. You're going to be hearing more about that later in this talk series. That hole in the middle, I'll fill in for you with a new tool called multi-channel individualized stimulation therapy, or MIST. This is an experimental tool which applies multifocal electric fields either with or without inducing seizure. The three tools on the diagonal were developed by our team. With MST, we can test model A that the seizure drives efficacy while the E field drives side effects. With TEST, we can evaluate model B that the E field drives efficacy while the seizure drives side effects. And with MIST, we can look at the interaction term. But before we dive into the new tools, let's see how much we can learn from conventional ECT. I've already told you that altering electrode placement from bitemporal to unilateral significantly reduces amnesia risk, but importantly, it achieves that while preserving efficacy when adequately dosed. I've already told you a 6-time seizure threshold is the key. This was a non-inferiority trial demonstrating comparable antidepressant efficacy on the y-axis between right unilateral ECT in the blue line and bitemporal ECT in the red. However, although changing electrode placement changes the distribution of the electric field, all three conventional electrode placements with ECT are inherently non-focal. The legend here tells this story. So this color bar shows you the electric field relative to the electric field threshold for stimulating neurons. The one on this field is this aqua color, so everything that's aqua or above is strong enough to stimulate the brain. And these red areas, which is most of the brain, are over 3-fold above threshold. We can draw a region of interest around the hippocampus, an area of the brain important for memory. And you can see that the E field in the hippocampus, with all three electrode placements, is above that one line. That's the threshold for stimulating neurons. So they all stimulate the hippocampus. Now that's just with a common brain, but let's look at actual patients' brains. So all of these patients with depression received the same dose of right unilateral ECT. However, what their brains saw was very different based on differences in their anatomy. Those individual differences mattered clinically. On the x-axis is the electric field in the hippocampus, and on the y-axis is a cognitive measure. And so it tells us that the variability of E field matters for cognitive outcomes, but we don't know with conventional ECT whether it's the E field or the seizure or both. But let's stay with the idea for a moment. It might be the E field. What is that E field doing? The Global ECT MRI Consortium asked that question by putting together almost 400 patients receiving ECT and measuring before and after a course of ECT the volume of the gray matter. And you can see here, these are effect sizes of volume increase across broad regions of the brain. So ECT increases this measure on structural MRI, and the degree of volumetric increase on the y-axis correlates with the strength of the electric field in each of those brain areas. Now that's correlation, but not causation, and we still don't know if it's the E field or the seizure or both that's resulting in these changes. And so let's go back to our new tools now and begin to unpack the mechanisms of ECT, starting with magnetic seizure therapy to test model A that the seizure drives efficacy while the E field drives side effects. So let's do an introduction to magnetic seizure therapy. MST refers to the induction of a seizure under generalized anesthesia and paralysis using a magnetic coil held on the head. Our goal is to achieve the efficacy of ECT without the same degree of side effects. The reason why magnetic induction might be more successful in doing this is that it avoids the impedance of the scalp and skull And we can target the induced electric field to superficial regions of the cortex, sparing deeper regions involved in side effects, such as the hippocampus. Here's an actual photograph of me performing the first, or getting ready to perform the first MST. This was done 25 years ago, and I'm standing next to Tom Schlepfer. We did this in Bern, Switzerland. Here's a abbreviated timeline of the translational development of MST. We had to redesign the TMS devices to be powerful enough to induce seizures under anesthesia. We did the first non-human animal MST in 1998, the first human in the year 2000. I just showed you that photo. We developed the first high-dose MST and did that in a non-human animal in 2004, and the first high-dose human MST we did in 2007. So now let's use MST to test the model that E field drives side effects while seizure drives efficacy. We published a series of studies in non-human primates and humans demonstrating that MST induces more focal electric fields. So B on the right here, that's the E field plot with MST with a round coil on the vertex. You see much cooler colors and more superficial than you get with right unilateral ECT on the left. We also published data in both non-human primates and humans that the seizures induced with MST were weaker and more focal. So this is topographical EEG with right unilateral ECT on the top and MST on the bottom. Cooler colors reflect weaker ictal power and less generalization. And we published a series of studies showing that MST has less memory loss. Here we're looking at the time to reorientation right after the treatment is finished, with MST in the hatched bars and ECT in the black bars. I'll just show you a few of these studies. This was our non-human primate study showing superior cognitive performance after MST in the blue bars compared to an animal model of ECT in the red bars. Green is anesthesia alone sham, so you can see that the green and blue bars are about the same. Here's serial list learning, and here's spatial working memory. In most cases, MST performing close to anesthesia alone sham. And then a first human study where we demonstrated that MST was safer. Here's again the speed of reorientation with MST in the blue, ECT in the red, and here are some verbal attention speed measures. Now, importantly, the comparator we used here was right unilateral ultrabrief pulse ECT, the safest form of ECT that is in conventional use. I'm showing you here still the largest to date randomized controlled trial to compare the antidepressant efficacy and cognitive outcomes of MST versus ECT. This is a between subject double masked study that we performed in 73 adults with a major depressive episode. And again, our comparator was 6 times seizure threshold, ultrabrief pulse, right unilateral ECT, because we needed to test whether MST was safer than the best that we could possibly do with ECT. Otherwise, what would be the point? Here's one of the cognitive measures. This is the time to reorientation after each treatment. The blue line is ECT and the orange line is MST. So patients were reorienting much faster with MST. This measure correlates with long-term retrograde amnesia. Here are some of the side effect measures on this radial plot. MST had less subjective side effects overall, especially in the physical and cognitive domains. So you can see here headache or nausea or dry mouth, muscle pain, and so on. Here are some of the measures of anterograde and retrograde amnesia. These were significantly reduced with ECT from before to after, and not with MST. And here's the autobiographical memory score, significantly reduced, pre-post ECT, not pre-post MST. And on this journal cover, you can see again here the E-field plot, and the coronal section nicely shows you how MST is really superficial cortex that's on the right, whereas ECT really penetrating deeper brain structures. Now, the big test is, did it work? Did it have antidepressant efficacy? On the y-axis here is the depression score, and the x-axis is after each individual treatment. We found no significant difference between the antidepressant response of ECT in the blue line versus MST in the orange line, and we did find sustained benefit at 6-month follow-up. The results of a confirmatory efficacy and safety trial of MST will soon be available. That study was powered to be a non-inferiority test of MST versus ECT, and we eagerly await those results to be reported out. But so far, we already have support from Model A that lowering the electric field lowers the cognitive side effects without sacrificing efficacy. But what if the opposite were true? Let's test model B using transcranial electric stimulation therapy. This is the brainchild of Bill Reginault, who published the open-label pilot data showing antidepressant effects of bifrontal ECT electroplacement in the absence of seizure. The test is done by using a standard ECT device with a bifrontal electroplacement, and it evaluates model B, that the electric field drives efficacy, whereas the seizure drives side effects. Like TMS, test focuses the electric field in prefrontal cortex, and like ECT, test uses high amplitude current, but it stops the train short of inducing a seizure. And this randomized controlled trial is currently underway. I don't have data to show you because it's still blinded, but we look forward to the results of that trial to see if they verify the open label data. Now let's go to the interaction term between the E-field and seizure with multi-channel individualized stimulation therapy. This is the brainchild of Z. Deng. The idea here is that you can use this E-field modeling not just to ask where did I stimulate, but to design where should I stimulate. So MIST on the right, you could define what E-field distribution you want and compute how much current to inject into different pairs of electrodes to achieve that desired distribution. We've built the prototype to do this multifocal stimulation, and you can see on this dummy head, it looks like an EEG cap, and indeed that's what it is. The electrodes are embedded into an EEG cap so that we can both stimulate and record. So it's integrated with EEG for seizure monitoring. This is a circuit diagram of the prototype that we've built. And here's a look inside the chassis of the actual device. So I'll draw your attention to the red square here. These are channel boards. A standard ECT device just has one of these because it's just stimulating 2 electrodes. However, in this case, we have independent control over 16 electrodes, so that needed 15 independent current sources. So it's like 15 ECT devices bundled into one. Now let me turn my attention to future directions. I gave you a spoiler alert that the conceptual model asking what is the role of the E-field and the seizure is incomplete because there's new mechanisms that are being discovered. For example, Zach Rosenthal and colleagues recently published in Nature Communications that after the seizure, there's another electrical physiological phenomenon that has been relatively unexplored with ECT, and that's cortical spreading depression. So it's a slowly developing depolarization that spreads like a wave across the brain, and this was demonstrated using optical imaging in a mouse model, as well as in a human model using non-invasive imaging. The impact of cortical spreading depression on the clinical outcomes of ECT, antidepressant efficacy, and cognitive side effects are yet to be explored and deserve exploration. And it'll be important to understand whether this CST phenomenon is being induced by the E-field or the seizure or both. I also told you that there are different approaches to titrate. In studies that we published, we introduced the concept of individualized low amplitude seizure therapy. The idea here is instead of using a standard fixed pulse amplitude, which is typically fixed at 800 or 900 milliamps, depending on which ECT device you use, we might be able to individualize the pulse amplitude to adjust for that variation in E-field. Remember I showed you that plot with many different patients with depression who were all getting different E-field strengths and distributions with the same current and same electrode placement? Let's adjust for that. The way we do with TMS, we adjust for motor threshold. If you don't adjust for individual variability, here's what you get. So this is a modeling study that we published where we varied systematically the brain volume, the scalp conductivity, and the skull conductivity, and modeled the same current amplitude across all of these models. And the red lines show you the significant variability in the electric field you get, which is on the y-axis, as we modulate these anatomical parameters. So instead, let's modulate the pulse amplitude to match each person's brain. And we can do that by titrating instead of by increasing the number of pulses, fix the number of pulses, and just increase the amplitude sequentially until a seizure is induced. When you do that, you find that you can induce seizures with lower overall amplitude. And what this does is it spares regions of brain from E-field exposure. So on the top coronal section shows you the E-field distribution with a conventional pulse amplitude of 800 milliamps. On the bottom is 200 milliamps. It's like turning a dimmer switch on the brain. You can get seizures with much less electricity induced by doing this amplitude titration. This also reduces the inter-individual variability in the dosage requirement. Here on the y-axis, we're plotting the coefficient of variation, which is how variable across individuals the seizure threshold is. If we titrated using the individualized pulse amplitude, which is in the blue bars, versus using a standard pulse amplitude, a fixed pulse amplitude to titrate seizure threshold in the red bars. Another interesting asset of amplitude titration is it can provide a strong means to predict individual dose need. How do we do that? Well, we do a motor threshold, in this case not with TMS, but with an electrical pulse. In this study, we titrated the motor threshold using individual electrical pulses applied under anesthesia on the y-axis and plotted how well did it predict seizure threshold. I'm sorry, on the motor threshold on the x-axis, how well did it predict seizure threshold on the y-axis? You see a very tight correlation. But even better than that, you might not have to titrate the motor threshold at all. You could model it. In this study in non-human primates, we simulated the electric field strength induced in the primary motor cortex, and that measurement also predicted the measured seizure threshold. So watch this space. We may see new ways to titrate and to better individualize dosage. And then the last few thoughts about future directions, other parameters to optimize. Frequency. What's the best frequency for ECT or MST? We asked that question in this study that was supported by NIMH, in which we parametrically varied the frequency on the x-axis and titrated seizure threshold with each of these frequencies on the y-axis. And you can see it's a U-shaped curve, both for MST in the blue line and ECT in the orange line. Conventional frequencies that we use to titrate with conventional ECT are on the inefficient part of this U-shaped curve. This curve suggests that it might be more efficient to use lower frequencies to titrate and to treat. And as you've seen earlier in this talk, any intervention that we do that lowers seizure threshold also tends to lower cognitive side effects, so it may be worth exploring. Another relatively unexplored parameter is the waveform. So I showed you these square waves that are delivered in bidirectional trains, but you can generate any sort of wave, biphasic, multiphasic waves, charge-balanced, prepolarization waves. These have yet to be explored in terms of their efficiency and their clinical outcomes. Now, MST is done by using a TMS device, but we already have the technology to modify pulse width with MST as well. That's called control pulse TMS or CTMS. With the CTMS device, which we published back in 2008, I'm showing you here the circuit a diagram and actual photograph of the first prototype that we built, you can generate near rectangular waves, you can modulate their polarity, all of these things can be done but have yet to be explored for optimizing MST parameters as well. What about directionality? I've shown you that conventional ECT is bidirectional, whereas MST is actually unidirectional pulses, but each pulse is multiphasic. In this study, we evaluated seizure threshold on the y-axis with unidirectional trains in the hatch bars and bidirectional trains in the black bars. Unidirectional was more efficient. Might be worth exploring. Lest you think these are all new ideas, they actually have been in the literature for over 40 years. This paper by Hiram and colleagues called ECT, The Search for the Perfect Stimulus, talked about trying unidirectional pulses, ultra-brief pulses, different frequencies. And back in 1985, they said that the device had been built and it's waiting clinical trials. We'd like to not have to wait 40 years for clinical trials for engineering innovations to meet clinical need. That's why I'm so excited to be the founding dean of the ASU School of Medicine and Advanced Medical Engineering, where we are training the future physician engineer entrepreneurs who will accelerate the translation of engineering innovation into clinical discovery and practice. Our graduates will leverage new tools such as artificial intelligence and data science to connect with more patients, to scale access to care, and ultimately to improve health outcomes. In 4 years, our students will receive both an MD and a Master's in Medical Engineering, and they will all do capstone projects developing engineering solutions to unmet medical needs. And we, pending accreditation, will be recruiting our first class in 2026. Now I'd like to transition to discussion and Q&A. And I'm open to any questions, but I've provided here a few discussion prompts should they pique any interest. What level of training should medical students and residents have on ECT? How can we address the stigma and increase access to effective treatments, including ECT? How can we disentangle this role of the different parameters, the E-field, the evoked brain responses, and mediating outcomes? And to what extent are the contributions different or shared across modalities of neuromodulation? I'm going to stop there and open it to questions. And again, thank you for the opportunity to address this group.
Thank you so much, Dr. Lisanby, for a truly outstanding talk. Very enlightening. Let's open the floor to participation. If you have a comment related to these discussion prompts, or a question for Dr. Lisanby, either type it into the chat, or if you would like for us to enable your microphone and camera to address the group, just raise your hand.
While we're waiting for questions to roll in, Dr. Lisanby, that was a fantastic talk, and I'm really curious to get your thoughts on how we incorporate some of those temporal dynamic parameters that you were referring to at the end of the talk, the pulse frequency as well as pulse duration and waveform into how we do electric field modeling going forward.
Yeah, that's a great question, Zach. So the E-field models that I was showing you simulated single pulses. They did not incorporate the temporal component. And the temporal component is of course important because we know, as I've told you, the physiological impact of a single pulse depends on its width, not just its amplitude. We're modeling its amplitude, but it depends on its width, and also the frequency and different training characteristics. So I think for such models, there have been some approaches to embed neural elements into the digital space to model how the brain responds to the stimulation. I don't think we yet have turnkey software solutions like SIBNIBs that incorporate those temporal components, but I see the field moving in that direction ultimately. That's going to be important. But I'd love to hear your thoughts. So maybe you have some ideas, Zach, or others on the line.
Why don't we turn the floor to, I believe Jarrod Ehrie raised his hand first. He will actually be our speaker on TMS in a couple months, and then Jeremy Bass afterwards.
Yeah, thanks, Yaakov. I just had a question about kind of other non-MDD indications for MST. Like I'm curious about kind of some research that's been done for things like schizoaffective disorder, catatonia, bipolar disorder. And I'm also curious, you kind of talked about this idea between determining whether E-field or seizure was kind of the primary mechanism of effect. Do you think it's probably the same for all these different indications, or might it differ between indications?
Yeah, thank you for that question. And for this brief introductory talk, I didn't do a complete survey of the MST literature, which I'm delighted to say has gone beyond major depression to look at some other conditions as well, including schizophrenia, schizoaffective disorder, bipolar disorder, and others. I stuck to depression because that's where we have the most evidence, but there are studies in literature that are beginning to look at these other indications. If MST retains the mechanisms of action of ECT, you might think it would have a similar therapeutic spectrum that ECT has, which is very broad. We don't know if that will be the case. It remains to be demonstrated. I think it's an empirical question. The side effects, though, I expect those to be preserved with respect to potentially having similar effects to ECT, but not having as much amnesia, because we see that in the non-human animals that we studied using a cognitive battery that was designed to be sensitive to the cognitive effects of ECT. These were healthy brains. They were not brains that were modeling a psychiatric disease. So I think that the mechanisms of the cognitive side effects might be preserved, but whether the mechanisms of the therapeutic effect are preserved remains to be seen.
Right, Jeremy Bass, I just allowed your camera and mic.
Thank you. Thank you, Dr. Lisanby. It's always great to hear from you. I was just going to bring up to the attention of those controlling the meeting. I don't have access to the chat. I don't know if others do too. So,
okay, I'll address that right now. Thank you.
But that was all I wanted to bring up. Thank you.
So we have a question from Robert McClure about how close is MST to routine clinical use?
So the CREST trial that I mentioned was powered as a non-inferiority trial with input from the FDA about what level of evidence FDA might need to see to evaluate safety and efficacy and substantial equivalency between ECT and MST. Depending on the results of that trial, we might be very close or we might be years away. It really depends on, we anxiously await those results being reported so that we'll know whether in an adequately powered study there is substantial equivalency. But that trial was designed to address that question.
Thank you. We have another question from the chat. What have you seen in terms of stigma with MST? Does it elicit the same negative reactions from patients or participants as ECT? If it doesn't have the same baggage as ECT, that alone would have value.
Yeah, well, thank you for that question. And addressing the stigma of ECT. So in the trials with MST that we performed, we recruited people who were referred for ECT and who were willing to have ECT. And so we didn't see the people who weren't referred for ECT, and we didn't see the people who weren't willing to accept a referral for ECT. I think that our field has a lot of work to do to educate the public about the modern science and medical realities of ECT today. While I'm hopeful that MST will be shown safe and effective in interclinical practice, I think it's also important that ECT remain in clinical practice as well, because there will continue to be patients who need it and who will benefit from it. So I will say, though, one benefit of MST is that it removes one of the barriers to acceptability of the treatment, and that is the memory loss. And the more barriers we can remove to accessing effective treatment, the better we'll be able to serve our patients.
Thank you very much. We have two more questions from the chat, and I recognize these names. These are both other leaders in the field. We have Sean Siddiqui and Linda Carpenter. I'm wondering from either of you if you'd be willing for me to allow your camera and mic so you could address the group. Linda Carpenter will be providing our talk on at-home devices. To start with, Sean Siddiqui will address us. Let me find him here and enable his camera and mic. Hi, Dr. Siddiqui.
Sorry, my camera's off because of this guy. Thank you, Holly. Awesome talk as usual. One question I've been wondering about in general is we talk a lot about volumetric changes with ECT and how they occur focally in some regions versus others. And that's been considered, like a lot of people talk about whether you can optimize electrode placement based on, to target the volume changes that seem to be more desirable versus less desirable. Do you think that's a logical leap, or do you think that we have data to support the idea that wherever you stimulate is where the volume changes?
So it's a great question, Sean, because targeting conventional ECT, even with a novel electrode placement, you're still going to induce a seizure. So you're doing both the E field and seizure. The data that I showed that show those volume increases It's after E field and seizure exposure, so we really don't know. But I think it's a scientifically useful approach to target the E field using electrode placement to target the E field to those volumetric changes that are associated with improved clinical outcome to see whether it makes a difference or not. But I think the best test, honestly, would be to do it with transcranial electric stimulation therapy. So give the E field without inducing a seizure. That would be the way. So one of the measures that we have in the test study is imaging before and after, because we want to know, is it the E field exposure alone, is E field exposure alone sufficient to cause these volumetric changes? So I think it's an interesting experiment to do. It will contribute to our knowledge of mechanism. Whether it will be therapeutically beneficial or not, that's why we do the experiment.
That's a good point. So I guess what I was wondering about more specifically was there's the intensity of the E field and there's this seizure, but there's also the location of the E field relative to the location of the volume change. It's plausible that where you stimulate is what changes. It's also plausible that the volume change is a compensatory reaction to something else that you stimulated. Do you know if anybody's actually tested that, or to see which of those is more likely?
Well, I showed a plot of correlation between E field strength and volume change, and that's on a voxel by voxel basis. So it really is where you stimulate that's increasing the volume, where you see the volume increases. But I don't really know how much the volume increase themselves will matter. They don't last. They're transient. A lot of them are kind of global. I think they are a readout of what the E field exposure was. I'm not sure that they're a mechanism, but that should be tested.
Yeah, I know that answers the question. It sounds like it does correlate. So if you want to target a specific sort of anatomy, you could. Whether you should or not is a different question. Right. Thank you.
Thank you, Dr. Siddiqui. And Dr. Carpenter?
Yeah, hi, Holly. Thanks for a great talk. That was excellent. So I've just heard a little bit lately about these studies that show that that anesthetic agents like dexmedetomidine and maybe propofol have these antidepressant effects when you give them at really high doses that do a EEG burst suppression state, like they really are deeply sedating. And I wondered if if that mechanism might be related somehow to the mechanism by which ECT works.
Yeah, that's a great comment, Linda. And, obviously, we know that some agents that are used for anesthesia can also be antidepressant, ketamine, for example, although that's at a sub-anesthetic dose. But it's an interesting field, and that might be another way of testing these models. They're not seizures, but this burst suppression is a physiological phenomenon. It would be useful to see really well-controlled trials on that intervention to see what sort of antidepressant or clinical effects it has, and also whether it has cognitive side effects.
Right.
I would think not because there's no E-field exposure, but we do know that anesthesia, certain types of anesthesia, certainly have some transient post-procedure effects. Yeah, it's an interesting field.
Thanks.
Thank you for that answer. We have another question in the comments from Dr. Richard Weiner, who's a psychiatrist and researcher in ECT. And it's a comment, E-field does not take into account pulse width. Would it be useful to look at something like charge per pulse field? And Dr. Weiner, you could type in the chat if you want us to enable your mic and camera.
Yeah, great to see you, Rich. Just saw you. And you're making a very good point. And yes, I think it would make sense to try to incorporate something more physiologically, or a measure that would incorporate more of the metrics that matter from the neuron's perspective. Very, very good point. And I'll take this opportunity to point out that Dr. Weiner is another pioneer in the field, trained me in ECT, and himself is an engineer and a psychiatrist, and really his career is a wonderful example of what you can accomplish by merging those two fields.
Any other questions from the group? This has been a very enriching conversation. Feel so honored to have so many pioneers and experts in one room together. Jarrod, I believe you had another question in mind.
Actually, I think they mostly got addressed. And with my question, I think in subsequent questions. I think my questions are done, but I don't know if there's anything else on your end.
Should we go to one of the discussion prompts, whether the attendees have any opinions about what sort of training medical students and residents should have in ECT?
That certainly feels like an apt topic for the consortium. We have a comment from Alexandria Hurley. It would be interested in hearing others' opinions on decreasing stigma as well. I know that's a very important topic within this field.
You know, I'll just add my 2 cents about the training issue, and I think it also relates to stigma. I think that as a psychiatrist, it's important to be able to refer patients that could benefit from ECT, whether you become an ECT practitioner or not. Being able to understand the risks and benefits and understand where ECT fits in the treatment algorithm for depression or other conditions that ECT is effective for requires some degree of training. And I think that the more training and more exposure can help you educate your patients about how to evaluate ECT as they consider if that might be appropriate for them. The more knowledge we have, this can help destigmatize. And the more knowledgeable we are, we can teach our patients and families about the medical facts, can help them filter through what is the sensationalized, fictionalized views versus the medically accurate reality of ECT.
Really appreciate that point. The chat section is exploding. We have a comment from Dr. Robert McClue wanted to weigh in. So let me find you and unmute you. Moment. Dr. McClure?
I just unmuted myself. Can you hear me okay?
Yes.
Okay. Turn on my camera too. Yeah, sorry for the mix-up. Just wanted to comment that medical students at UNC and University of North Carolina, Chapel Hill, I mean, all the medical students who rotate through their psychiatry rotation will see an ECT procedure, and many of them also a TMS and esketamine.
Yeah, thank you for that. And my experience also as a medical student at Duke, seeing ECT firsthand as a resident with a leading expert, Rich Weiner, to be exact, not only shaped my understanding of ECT, but influenced my decision to go into psychiatry. That this is a field that can bring about very rapid response for among our most severely ill patients, and seeing that as a medical student is a very powerful and important learning opportunity.
We have a couple comments about the process at various folks' institution. Food Chai comments that at UCLA we offer ECT exposure for medical students and an elective for neuromodulation residents, an elective for neuromodulation for residents. As far as they know, geriatric psychiatry is the only fellowship with built-in ECT training. Dr. Jeremy Bass comments. I received it as a resident because I requested it. For those that did not request, they did not receive. And then we have a couple opinions from the group. Dr. Jason Z, one of our attendees at Columbia, believes exposure should start in medical school, hands-on experience preferably. And Seizure Wilson comments that they think all trainees should directly observe the course of an ECT patient. We have some other comments here from Zach about the relationship between hands-on experience and reducing stigma. Jeremy Bass comments that there's also the issue of rules at the state level. The two largest states by population have some of the strictest rules around it. Anybody who's commented, feel free to raise your hand to address the group. There's some other comments about the process at Washington University, where residents basically run the ECT service, very engaged. Ailani, one of the consortium leaders from Maimonides, comments that it's important to have both didactics and hands-on training for ECT residents, whether or not they will integrate this into their future practice. Becomes particularly difficult in certain patient populations and settings like in community hospitals. Dr. Chaikin comments that anybody interested in the topic of ECT, the ISEN Education Committee is working on curriculum recommendations and standards for residency training. But the reality of having different access to ECT at different programs poses a challenge. A lot of gratitude for Dr. Lisanby in the chat. Opportunity for me, maybe for one. So we have about 1 minute left. So any closing thoughts, Dr. Lisanby?
I really appreciate all the great questions and having the opportunity to share. And this is, a rich area of science. It's important clinically. It's an opportunity to really bring the latest medical advances to the bedside, literally, to benefit your patients. And so, thank you for this opportunity.
This was an incredibly exciting and enriching talk. Thank you again. This is what the consortium is all about, having leaders come and speak with trainees and faculty members, us to learn from one another, bring this material alive. We couldn't be more grateful to have you here, and for everybody who participated. We hope to see you next time in September. Thank you all.
Thank you.